So let's start discussion from the notes. We will discuss it in easy way, in interactive way. So let's start. So here we will start from the topic enzymes. Is my screen visible to you? Tell me. Can you see my screen? Okay. So what is the name of your topic? Can you clearly hear me? Okay, fine. The name of our topic is what? That is the enzymes. So enzymes are what? As I told you already, they are called as biological catalyst. So why enzymes are called as biological catalyst? Catalysts are what? Catalysts are those substances made by the living organisms. They are biological catalyst. Why I am saying biological catalyst? Here the question arises that why I am saying term biological catalyst. Bio means what? Bio means living. So they are the substances which are produced by the, or I can say made by the living organisms, and in turn they are going to speed up the chemical reactions without being used up or permanently changed. Okay, they are not used up in the reaction. Enzymes are not, I can say enzymes are not consumed in your chemical reaction, or there is no any permanent change in your enzyme. So I repeat again, what are enzymes? They are the biological catalyst. So what I mean to say by the biological catalyst, they are the substances made by the living organisms that speed up the chemical reactions without being used up or permanently changed. They are not used up in the reaction, that I can say they are not consumed, or they are not permanently changed in your chemical reaction. So, in simple words, what I can say that enzymes make life reactions happen faster. Obviously, reactions can take place in a very less time. Reactions can happen faster and also easier. So, this is the importance of your enzymes. Now, here the question arises that why do we need enzymes? So, all the living organisms perform thousands of chemical reactions every second in our body also. You know that many chemical reactions they are being taking place. In fact, I can say thousands of chemical reactions are taking place in my body in this time, right? And so, for all those activities, for all those chemical reactions, means those chemical reactions should happen fast. Okay, so for that purpose enzymes are there. Okay, like breaking down food. For this is the process of digestion. For this enzymes are needed. Building new cells of your body, right? Building new cells of your body in case of cell repair, it is needed. In order to release the energy, that is by the process of cellular respiration, we know that ATP, which is the energy currency of cell. Okay, so energy is released. Repairing tissues of our body, we know that tissues are made up of what? Cells. Group of similar cells, they are called as, means they form the tissue. So repairing tissues also the function of your enzymes. Okay, so these reactions are too slow to sustain enzyme, to sustain life. Means these all reactions that I discussed here, these reactions are very very slow if enzymes are not there, but they are going to sustain your life. But now important concern here is they are going to sustain your life. So enzymes make all these activities fast enough for survival. So this is the importance that we discussed here. Okay. So now I am going to explain here the properties of enzymes. Our enzymes are made up of what? Proteins. Enzymes are made up of proteins. Some have some have a non protein part also. This is important. Some enzymes have a small non protein part also, but generally all enzymes are made up of proteins. So but protein is there, but there can be a non-protein part also in some of the enzymes. But main thing we have to keep in mind, all the enzymes are proteinaceous in nature. Next point, speed up the reactions to lower the energy needed. So they lower the energy needed for a chemical reactions to take place. Which energy? Activation energy. Enzymes, they work by lowering activation energy. Enzymes, they work by lowering the activation energy. Okay. So due to this reason, your reaction can take place more quickly. So, what is the activation energy? This you can write down. Activation energy is the minimum amount of energy. It is the minimum amount of energy needed. It is the minimum amount of energy needed to start a chemical reaction. It is the minimum amount of energy needed to start a chemical reaction. So they lower the energy needed. Which energy they will lower? Activation energy. So reactions occur quickly, or I can say with a more speed. They will become fast because obviously they are going to lower the activation energy. Next, enzymes are specific in action. Each enzyme works on only one type of molecule. Enzymes, they have a specific action. Very interesting that each enzyme works on only one type of molecule. Example, amylase works on the amylase is an enzyme. Amylase is an enzyme that have a role in carbohydrate digestion. So it works on the starch. Starch is what? Starch is a carbohydrate actually. Lipase enzyme have role in the fat digestion. So lipase works on the fats only. So in this way we can see that each enzyme has a specific action. So this is called as enzyme specificity. Next, needed in small amounts. Yeah, human case, like in case of, yeah, amylase enzyme that have role in the carbohydrate digestion in our body. Lipase enzyme have role in the fat digestion in our body. So like our DNA ligase is an enzyme that we study in the biotechnology portion. So DNA ligase joins the broken DNA fragments. DNA ligase joins broken DNA fragments. So obviously, if it is going to join the broken DNA fragments, means a bond is going to form here. So by forming which bond? Phosphodiester bonds. By forming phosphodiester bonds. And also enzymes are needed in very small amounts. A very small amount of enzyme can handle the large amount of substrate. So what is substrate? Substrate is the substance on which enzyme acts. Substrate is the substance on which enzyme acts during a chemical reaction. For example, starch is the substrate for starch is the substrate for amylase. Starch is the substrate for amylase. And also proteins are substrate for which enzyme? Protease enzyme. And in the same way, fats are the substrate for fats are the substrate which enzyme? Lipase enzyme lipase. Okay, so enzymes are needed in small amounts, that I told you already. Not used up. Enzymes are not used in a chemical reaction. They remain unchanged at the end of the reaction and can be reused. They can be reused, as I told you already. Works under optimum condition. A particular optimum temperature is there, optimum pH is there. So they work at the particular temperature and pH. So which is called as the what? Which is called as temperature? Yeah, tell me. Optimum temperature is the temperature at which the enzyme works the fastest. Like for most of the enzymes in the human body, the optimum temperature is 37 degrees Celsius, which is the body temperature. If if there is if there is low temperature, then the enzyme activity becomes slow. And if there is high temperature, like above 45 to 50 degrees Celsius, then enzyme become denatured, or I can say enzyme become destroyed, and the and it will stop the working. So optimum pH is the pH level at which the enzymes work fast. Like for example, pepsin enzyme that you will study. Pepsin enzyme that you will study, right? It also works best at a particular pH, like nearly pH of 2, which is what? Strongly acidic pH. This is what? Strongly acidic pH. So, pepsin enzyme in our stomach, it has role in the protein digestion. In our stomach, pepsin enzyme help in the protein digestion. So enzymes work best at optimum temperature, usually 37 degrees Celsius in humans, and each enzyme has its own optimum pH. Whether it can be acidic, whether it can be neutral, or it can be basic. It can be acidic, it can be neutral, or it can be basic type of pH. Now next is they are sensitive. Enzymes are sensitive. High heat or extreme pH. As I told you that enzymes, they work best at a optimum temperature and pH. But if there is high heat or extreme of pH, then that can destroy, or that can inactive the enzyme. So this is important here. So these are some of the important features we discussed for the enzymes. Okay, fine. Next is the enzyme terminologies. So enzymes, they act as a catalyst, I told you. Like amylase enzyme that have role in the carbohydrate digestion. Like protease enzyme that have role in protein digestion. Next, we have substrate. As I told you, the substance on which the enzyme acts, that is the substrate. For example, starch is the substrate for amylase enzyme. Next terminology that you will study here, that is the active site. Active site is what? That is the specific region on the enzyme where the substrate fits. Like I can show you here. Here I can show you this thing. Active site. Active sites are what? That is the region of the enzyme where the reaction takes place. So active site is the region of the enzyme where the reaction take place. The substrate molecule interact with the active site and then undergo a chemical reaction. So what is active site? It is the reason, or I can say it is the part, right, of an enzyme where the reaction takes place. So substrate, you already know what is substrate. So our substrate molecule that interact with the active site of the enzyme, and in this way it will undergo a chemical reaction. What is the product? It is a substance formed after the reaction. Yeah, great. So, what is product? Product is the substance which is formed after the reaction. So, these are some terminologies that we discussed for the enzymes.